So first of all, I would like to thank the organizers for inviting me here. It's kind of nice to be able to present my work to you all. This presentation will be much more applied than the previous one, but I hope you can still appreciate it. And in particular, we're going to be talking about molecules and different way we can represent molecules, either as voxels or narrow fields, to perform struct condition molecule generation. And if you don't know what voxels and narrow fields are, those are terms that come from the computer vision community. And I hope at the end of the talk you can at least understand what they mean and how we can use them. So first of all, why we want to do molecule generation? Well, one argument is that pretty much everything in the world is material, and everything in the material world is made of molecules, maybe with the exception of dark matter, but I guess we don't care that much about that on applications. And if we're able to actually succeed on generative models for molecules, you're able to generate molecules with properties that we care, we can then have impact in many different applications. It's like drug discovery or biology, materials, the environment, and so on. In particular, I will be focusing more on drug discovery because one, I found it a very interesting application where the goal is to try to cure diseases in a way or another. And also because that's what my employer do, so it's kind of easier to convince the research on this aspect. But here we're trying to thinking about representations for molecules that are a bit agnostic to the type of molecule. So in theory, what I'm talking here could be applied in all of different modalities. We just have to switch the data that we'll be working on. So first, how people are doing molecule generation these days. So the status quo on this field is to consider molecules as point clouds of atoms, where each point represents an atom type, and then it also has its XYZ coordinates in space. And generation is usually done with a diffusion model or something similar, usually a score-based model, where first we sample points from a known distribution, let's say Gaussian, and then we apply the reverse diffusion model that has been trained to move those points around and also change their atom types as the reverse diffusion process goes on until it's over, and hopefully we're going to end up with a nice conformation 3D structure for a molecule. So this is nice because people can leverage SC3 equivariance GNNs, and especially useful when you're in a low domain of data. And also those models has been applied to many different applications. For example, conditional generation. Let's say we have a protein target that we're interested in, and they would like to generate some molecules that would bind to that particular protein target. This is actually going to be the topic of the presentation. But also people applied those models to small molecules or to proteins as well. This is what we've seen in the first presentation, David Baker's work that ended up getting a Nobel Prize in chemistry. And also people are now trying different generative models like flow matching, which is almost the same as diffusion, but it gives new papers to people. And what we decided to do is to go against the status quo, and this is a little bit what it's linked to the previous presentation, at least on the title, is that we're going to use as a presentation in which we don't necessarily have SC3 equivariance, but however, we can leverage much more expressive models. And then the question is does this help in the task of molecule generation? So we decided to represent molecules here as fields or molecular fields, which are basically functions that would map points in space into a vector of atomic densities. So those representations have been used before. In fact, when people start using deep learning for molecules, they start using those kinds of representation. So they have those densities, they discretize it into space, and then they use what people have been using in computer vision. But it's never been very successful. So everyone switched to point cloud representations. And it also has never been not been successful in generative model. So we decided to revisit this idea and actually see what we could do with this. So the way we represent molecules, basically you can think each atom has its atomic density is more or less a 3D Gaussian blob in space, where the maximum value would be at the center of each atom, and then it will decay exponentially as we move away from those atoms. And then at each point in the space, we would integrate the density of each atom, so that then we have our function. And in particular, we use this formula here to compute this density. So we have this function has some properties that are going to be useful, as I'll tell later. It has the maximum value at the center of the atoms, and then it decays as we go far away from it. And we also model one atom type per channel. Similar to images, we have the red, green, and blue channel. Here we're going to have one channel for each atom type that we have in our data sets. For example, one channel for carbon, hydrogen, oxygen, nitrogen, and so forth. So there are the most common way that, the most trivial way to consider those representations is to use voxel grids, which is basically we take like this molecular field, density field, and then we discretize it into the 3D space, generating like this voxel grid. So voxel comes from voxel element, which is like similar to where pixels come from. Pixels stands from picture elements. So this is basically like an augmentation to 3D of the 2D images. And because of that, we can usually pretty much the same models that one can use for computer vision, just like out of the box. And then another one is those representations called narrow fields, which the people also call it like implicit neural representations or coordinate-based neural networks. So in this case here, we consider molecules as continuous functions in space. We totally pass the discretization step of like voxel grids. And those molecules are modeled implicitly with neural networks. That is where the neural from neural fields come from, actually. Yeah, so this is nice because it's a very compact representation, so allow us to model like pretty much with the same architecture and with the same generative model, larger molecular systems because we bypass the restrictions of like voxel grids related to like memory and computation costs. Okay, so for the rest of the presentation, I will talk to you about how we can use those two different representations, which are quite popular in computer vision, but apply to molecule generation. Yeah, so just as I say, this is highly inspired by vision in terms of representations, in terms of the architectures we're using, and in terms of the generative models that we're using as well. Also, for the rest of the presentations, I will be focusing mostly on this problem that people in the community call structure-based drug design. So here the idea is to try to design molecules that would bind to a specific 3D protein structure with high affinity and high specificity. So, if you abstract enough, we would say like, okay, so given a protein pocket, we would like to have a model that would just like spit out a bunch of like ligands or a bunch of molecules that would hopefully bind to that particular protein pocket. And this is just an example of actually the generative model that we designed that are like just spitting like a bunch of candidates, some of them good, some of them bad. Okay, so now in the first part, how we can use like voxels, voxel grids to do conditional generation in this setting. So this problem, can be translated in machine learning language. Basically, has to do with 3D molecule generation, conditional and protein target. So it's a conditional generative model where the conditional signal is a protein target of interest. So this has been starting to get attraction in the community, but it's a very recent line of research. It started more or less in 21 or 22. So people have trying like voxel grids. Actually, it was one of the first works, but unfortunately, that did not work at all in practice. And then people moved away from that and started using point cloud representations, either using like an autoregressive approach, which is going to be sampling like one point or fragments at a time. And now more recently, people are using interdiffusion or Bayesian flow nets on those point clouds, or also like flow matching, which is like how I explained in the beginning how that's usually done. So here what we're doing, we're using like a different generative algorithm, which it's called like walk jump sampling. And here I'm just going to tell you very quickly like the intuition of this algorithm. So it is a score-based model. So in a way, it's very similar to diffusion, but in other ways are very different than diffusion. But the way we train those models are very similar to diffusion. So pretty much everything I'm going to tell you in this presentation can also be applied for diffusion models. So the idea is we want to sample from this like very high dimensional space of molecules. So sample from X from P of X. But that's like very difficult because it's like an intractable problem. It's very high dimensional, and the probability mass like is concentrated in a very low dimensional manifold. In fact, this is so difficult. That's why all the research and like deep generative models came from. It's try to be able to cope with this problem that is just very difficult to sample from high dimensional spaces. So the intuition of like walk jump sampling is the following. Even though it's very hard to sample from P of X, if we smooth this space P of X, basically like if we just like convolve it with a Gaussian kernel with like a fixed noise level, or in practice, we just add a bunch of noise to our data, we smooth out the space, then we are not like in a low dimensional manifold anymore, but we're like occupying all the ambient space. And this makes it like much easier to sample. However, it's kind of useless to sample like those noisy objects. And then like the idea of like walk jump sampling is like we first sample from these noisy objects, and then we estimate the clean images given like those like noisy ones. And the hope is that like those two-step process would be easier than to do just one step. Again, just to repeat myself a little bit, it's going to be hard to sample from P of X, but it's easier to sample from P of Y. So the idea, that's where the name walk jump comes from, is that we're going to first sample Y from this P of Y, which is the walk step, and then we're going to do the jump step to estimate X, the clean objects from Y, the smoothed one. And the walk step is done with a Langevin MCMC. And to do that, we need the score function of this smoothed P of smooth distribution P of y. And then we estimate x from y with a minimum mean square error estimator. And in practice, as usual, we're going to be using a denoiser. And it turned out there is like this famous equation that people are now calling like 3D Miyazawa that dates back from the 60s that link those two different objects, like the score function and the minimum mean square error object. So in practice, all we need to do is to learn a denoiser, because with a denoiser, we can compute the score function of P of y, and we can have this, be able to estimate the x from this like smoothed y. And then in the conditional setting, it's pretty much the same idea. We just need to change the x, the P of x to P of x given xc, and P of y to P of y given xc. So xc here would be like the conditional signal that we are considering, in this case, the protein target. So yeah, so everything behaves the same. We still do like this walk jump step, Langevin MCMC on the first one, minimum mean square error estimator for the second one. And then what we need is a conditional denoiser in this case to like be able to do those two steps. This is how we do, and this is where the influence from computer vision comes from. The architecture that we use here is exactly the same as what people are using in diffusion, like those very nice models that work very well in practice for 2D images. The only difference is that like our objects now, they are like 3D tensors instead of 2D tensors, but otherwise it's pretty much the same thing. So we start with our data, we would contain those complexes of the pocket and the ligand molecule. We voxelize each of them individually, and then we're going to train this conditional denoiser. So we're going to be adding noise to one of the ligand, which is the molecule we want to generate. We don't add noise to the other one, to the pocket. And then we're going to train a model with an L2 square loss, just like actually a denoiser loss, to try to reconstruct the original ligand that we added noise to it. So it's very simple. It's very like straightforward as well. This is what people have always been done. The only difference here, we actually use like the statist of the state-of-the-art architecture from computer vision. It was published last year by some people from NVIDIA. It's called EDM2. We just basically get that architecture, modify it a little bit. So instead of having 2D kernels for our convnet, we just have like 3D kernels instead. Even the self-attention layers that also exist in this architecture remains untouched. So it's a very minimal modification. But the idea here is like if we train this in a proper way, and the denoiser is actually good enough, then we can approximate those two objects that we need to do walk jump sampling, which is one is the score function of P of y, given xc in this case, and the other one is the minimum mean square error estimator. And yeah, so an expressive neural network is like key here. We really need to have like very expressive architecture because we don't even have any equivariance here. So the hope is that having a powerful architecture, and if you're able to train it like properly, we can do the sampling. And this is how we do the sampling, actually. So we start with like an initial like protein target. Let's say some biologist or chemist comes to us and say like, hey, this protein is important for this disease. We need to find like some molecules that will bind to it so that we can, switch from the cellule from like the disease state to the health state. And the way we do is we start with this protein, we voxelize it, then we initialize our chain with like noise, let's say Y0 here. And then we just do like a Langevin MCMC. So we just start, this is the walk step. We start walking. First we start Y with noise. We have some warm-up steps that we go from the noise to the smoothed manifold. And then we start sampling it with the Langevin. And for that, we need the score function. In practice, we use like not the simple Langevin version, but use the underdamped one. But in theory, it's more or less the same thing. And then at arbitrary times, while we're like sampling this Y space on this moved, we can do this jump step. We can do it like any time we want, after 10 walk steps, after 20 walk steps, or after 100 walk steps. It doesn't really matter. At arbitrary time k, we do this jump step, which basically we denoise those like smooth objects to get the clean ones. And then one last step is like once we have those molecules in those voxel grids, for us to communicate this to computational chemists, we need to give them like molecules per se. So we have to transform those voxel grids into molecules. And the way we do this is like with like a simple post-processing step. So we start with noise. From walk jump sampling, we sample like some voxel molecules. And then we just find the peaks of those voxel grids, because remember, by construction, the center of the atoms are the maximum values, which are value of 1. So if those voxel grids are good, we're going to be able to find like all those atom coordinates. We do some optimization, so we move those points around a little bit to better fix the grid that has been generated. And then once we have that, we have like a point cloud of atoms, just like every other method that operates on point clouds. And then from there, we use like a chemoinformatics software just to find like the bonds of those molecules. And then finally, we have a molecule, and we can do anything we care with that. In particular, give this to computational chemists for them to assess the quality of those molecules. So here are just some examples of like this walk jump sampling in practice. This is like some targets from the validation set. And in each of them, we are doing like 20 walk steps and then one jump, 20 walk steps and a jump, and so forth. So it's kind of nice because if you see like, if they are close in the number of like walk steps, the structures of the molecules are very similar. But then as we keep walking away, the structure starts to become different. So locally, like they're very simple, the modification is very small, but it keeps changing as we walk through this chain. And what it's nice is because, you know, it's very difficult to do MCMC on like high dimensional objects, but because if we smooth, those chains, they can run for like 10,000 or even try like 50,000 steps and it just don't diverge. It just keeps sampling, which is like kind of interesting in practice. I will skip this for the interest of time. It's just like a bunch of metrics, but you can take a look at the paper if you're interested. There's like many more metrics than those on the paper. The TLDR is that like the model we propose performs better than like all graphical models, at least when we publish this paper. And it's also much faster to sample. Well, one thing that needs to be taken into account is like all of those in silico metrics, they're like very bad. If you talk to computational chemists about them, they would just say like, no, this is not good. But you know, if you have like algorithm A that is better than algorithm B in 15 out of 20 bad metrics, we can maybe argue that one algorithm is still better than another. We train like this model in like much more internal data because for here we just train on publicly available data so we can benchmark with publicly released models. But I also train it in a lot of like internal data. And we are actually starting to try to synthesize molecules with this model. We just have like some very preliminary results at this point. We're finding like weak binders, which is not great. But on the good hand is that like it's been better than all the other models that has been tested, either based on deep learning or based in more like classic computational chemist approaches. So it's nice because it works very well on small molecules, more or less at least. But it's hard to scale because, you know, like we discretize the space here. So so we discretize the space. So we can't really like grow too much that box that we're modeling because it grows cubically with the dimension, you know, with the grid length. So we are doomed. We need much more, cubically more memory GPUs to be able to scale this to large objects. And this is what motivates us to have another representation which is more scalable, which is to instead of consider molecules as voxels, we're going to consider them as like narrow fields. So, and now I'm going to tell you how like we represent those molecules implicitly with those as functions or fields, and how we use that to generate molecules. Last year, we worked on the unconditional generation version of this. Like we're just sampling molecules unconditionally, just trying to match like a distribution of a holdout set. And now currently, we're like extending this to like structure condition version of it. So same problem as before. We have like a protein target, and we want to generate molecules that would fit to that, but using this different kind of representation, which, by the way, is now the state of the art in 3D computer vision. And like most of the people now in that community are using this kind or variations of this kind of representation. So remember like that molecular fields are like functions that map like points in space into vectors of atomic density. And the narrow fields are basically neural networks that approximate that molecular field. And in practice, the way we're going to be thinking about this is that the narrow field would be like a representation that can be seen as like a latent representations for molecules. And then we're going to have like a latent generative model on the top of those narrow field representations. So to model like a full data set of molecules, what we do is that we condition those narrow fields on a latent variable z. So we use what the people in the community call like this auto-encoding approach. So we introduce like an encoder architecture that will take a molecule and then generate this latent space z. So the representation of the molecule here could be anything. Could be point clouds, could be like a string representation. We are using like a very low resolution like grid. And then as the encoder, we are using a 3D CNN. But those things can be anything. But once we generate this latent space z, then we're going to like pass it forward through our like conditional narrow fields. We're going to take like the z as an input, but also like points on the XYZ space. And then what we would like to do is to train a model that, given the z that would represent a certain molecule in our data set, and give it like points in space, we would be able to represent its like atomic density like properly. So the way we train this is with like supervised learning, basically. So at each like iteration, we're going to sample a new molecule. We're going to add some rotation to it because the model is not equivariant. Then we're going to sample a bunch of points in X. This is like a continuous space, so we cannot like take them all at once. So just at At each iteration, we sample like a bunch of points, I think 2,000 in practice. Then we forward the latent Z and the points throughout our neural field, and then we would like train the model to reconstruct the actual molecular field. Then, you know, like if we do this like a lot of times, hopefully the model will train well, and then we are able to just reconstruct all the neural fields of every molecules in our data set. So if we abstract a little bit here, this can be seen like pretty much like a latent representation for molecules. Now, instead of reasoning on that voxel space that we had before, or the point clouds, which what people did before, we are reasoning, or like the generative model will be operating on this latent space Z, which is much more compact, even though we're able to model like much larger like volume. So again, abstracting enough, this is almost like an autoencoder. We start with the molecule, we learn the latent space Z, and then we decode with this fancy neural field encoder to like the molecule that we would care. And then we train this in both structures, both the ligands, so both the molecule we want to generate, but also the pockets, the proteins in which we're conditioning the model on. And then just as before, we learn a conditional denoiser here, but instead of the conditional denoiser being applied on the voxel grid, the 3D space, we apply this in this much more compact Z space, so this latent space that's been learned through this neural field representation. This is very similar to how people do generative models on images now. Like everyone moved away from operating on the pixel space, and now everyone operates in a latent space. So every state-of-the-art model now, they just reason or they just operate on a latent space, just because it's more efficient, and especially in this case of molecules, we can like model like larger molecular systems. On the paper, we can model very large molecular systems, but like we're just growing like step by step because it gets kind of harder computationally as we keep growing the space of the box that we are modeling. OK, so the way we do sampling is exactly the same as before, except that instead of sampling the voxel grid, we sample that latent space. And then once we have this latent space, we then need to decode the molecule from it. So there is also another post-processing step here. So we initialize, so we start with a pocket that is given by, let's say, a biologist of interest, and then we, instead of voxelizing it, we generate this latent space Z. And then we initialize the noisy latent with Z0, and then again, we do those walk steps with Langevin MCMC, just exactly the same as before. And then at arbitrary time, we do the jump step to clean up those noisy latent codes to have like a clean code. Now, with that clean code, we just need to actually extract the molecule from it in a way or another. So there is like a post-processing step that would convert the code that has been generated into a molecule. So then again, we can communicate this to biologists or chemists. Yeah?
Process, do you work with a fixed noise level sigma or different noise level?
Yeah, so here we're using fixed noise levels. Right now we're exploring to do like a mixture of diffusion and this approach in which the denoiser is like multiple steps. I think it's almost magical that this works with just a single noise step. I think on the voxel grid, it kind of makes more sense because the signal is very sparse and very like low frequency. It's just like blobs in space. So I'm not that surprised that one step denoising would work there. But here is a latent space that we don't really have much control. The only regularization we do when we learn this latent space is to do like this VAE regularization, you know, with the KL regularization. But that's all. So it's, the Z space is also continuous. So intuitively, it should not really work with just one single denoise step. But in practice, it does. And we don't understand very much well. But we also believe that if we have like multiple steps, either as a diffusion or a mixture of this approach and diffusion, the results will just be better. So that's why we're exploring that. The problem of diffusion is that it's much harder to tune. So it's still not working, but we have colleagues working on that. Yeah. So then the post-processing is very similar as before. So we have the latent code Z that we generate. And we just render like a very low dimensional grid, basically with like a for loops, scanning like the XYZ coordinates to come up with like some low resolution voxel representation. Then we find those peaks, which is like basically the local optima of this box. And then with that, we just do some coordinate refinement, and then we have our points again. And then with those points, we are in the same situation as we are in point clouds once again. And what we do is like we get the chemoinformatics software to just find the bonds based on the distance of those point clouds and the identity of those atoms. We trained this model in different data sets. One is of small molecules. Another one is like antibody CDR loops. So CDR loops are like the regions of the antibody that are important for binding into like a certain protein. So this is like what antibody engineers and like what people in the field care the most. So we try to first work with just CDR loops only, but the idea is to increase this to like larger spaces of the antibody. And then also another modality which is called macro-cyclic peptides. This is a modality that people in the drug discovery space are getting super excited about because they kind of share some properties of small molecule drugs and of like large molecule drugs. Those are just some statistics of the different data sets. As the name implies, small molecules are quite small. They on average are 24 atoms per molecule. And then, the other ones have like it's much bigger systems. And VoxBind, so the previous model, the one that operates in voxel spaces, can only work here because, the other ones is just, the box would be too big and we need like too many GPUs. I mean, or maybe like 20 more years of GPU technology to be able to model those big spaces. This last data set is not published as internal, but we plan to release because they're built just on like the PDB data. So once we release this work, hopefully we're going to be releasing this macro-cyclic peptides data set as well, because it's something that really does not exist in the public domain. One thing that is interesting that we do here is that we train one model on all of those modalities. So instead of having like one model per modality, because our model is, we don't have any like domain knowledge or not much inductive bias, but on the model, we can just model anything. Everything is just like atoms in space with this weird blob representation. So we train the neural field with all of those representations, small molecules, CDR loops, peptides, and all of it together. So we just learn a very general like neural field. And then we also train our conditional denoiser on all data. But now we have to actually condition it on the modality as well. Because in practice, given a target, we would like the model to generate just small molecules or just CDR loops or just peptides. So we add one extra conditioning signal there that actually would condition into what modality of molecule we would like to generate. So this field is very general. So here we're just training for now only on those 3 data sets. But we would like to train in all the structured data set that exists, including data in materials and enzymes and all kinds of proteins and everything. So that's kind of a to-do for the future, to just have a model that would just generate any kind of molecule. That would be the dream one day. These are just some quantitative results. The model is working on all of those modalities. We didn't have we didn't do a lot of benchmarking with public models yet. That's what we're currently doing now. On small molecules, we're benchmarked already because we already have all the code base and everything. So this new model, which we're calling FuncBind, having similar performance as voxbind, which is it's around the state of the art on this public benchmark. For the antibody CDR loops, we only tested on internal benchmarks, but the results were good. So we are actually now designing some molecules from our model. The designs are already out, but we don't have the results yet. So all we have is in silico predictions of what those designs would be. And it seems to be good, but we have to wait until we actually get the wet lab experiments to have any conclusions. And then macro-cyclic peptides is a very new data set. We're going to actually release the data set. So there's not really much to benchmark at this point. We would love to be able to synthesize molecules on that peptides modality too, because then we're synthesizing on all the modalities that our team are interested. So that would be super cool. But at this point, we didn't find any use case in practice to synthesize macrocyclic peptides. And with this, I conclude. We propose new ways to represent molecules that are different than the status quo, which is point clouds. We don't add any priors on any molecule structure. The molecules are just we just model the full box. It could be as many atoms as we want, any modality. We don't have SC3 equivariance, but instead we have a much more expressive model. And then we play with this trade-off, and we show that a more expressive model can be good if everything else is on place. This model, because we don't have any domain knowledge, can be applied to different data modalities. As we've shown here, it's already working on small molecules, peptides, and proteins. But we would also like to maybe someday, if we could, to apply this on RNA and DNA structures as well. And also, it's agnostic to the data type. Right now, we are using those molecular fields, but you can think about applying those models into electrodensities and actually have colleagues that are trying to do that as well. Or if you consider just the surface of the proteins, or even the quantum mechanics orbitals and stuff like that, the model is agnostic to whatever. As long as it fits into a box, in theory, that should work. And this is an all-atom modeling, again, saying what I said before, which is kind of different than what people usually do, especially when they model proteins. They usually try to do some kind of coarse graining or just model the backbone. But in this case, we just model all atoms and independent of its modality. So as a next step would be to finish the benchmarking of this latest model that I told you. and then also scale up this to even larger systems. So we start with like very small systems, just like this QM9 drug when we first started those projects, which are molecules that had like up to 9 molecules, sorry, up to 9 atoms. And then we're like slowly like increasing, and the idea is to like keep increasing and see when we like hit a wall, because so far we haven't, but very likely we will at some point. And with that, I would just like to acknowledge my colleagues that helped on like all this work. And with that, yeah, I'm done. Thank you.
So for the sake of time, we'll take one question. Speaker's choice.
Oh, sorry. I don't know. No, not speaker choice. That's too bad. I can do that.
For the talk, nice result. Can you clarify why this representation is more expressive than the traditional one?
Than the point cloud one?
Yes.
Yeah, it's because the point cloud operates in graph neural networks, and you know, graph neural networks tends to like lack a bit of expressivity compared to, let's say, MLPs or transformers or CNNs.
Why? I mean, you can just have universal approximation, right, under equivariance.
People have shown that in practice. I think there are some papers that show that. I think on the top of my mind here, I cannot explain you like technical details, but I would say it's known in the literature, and it's in practice that's what we observed as well.
So in practice,
the thing is, the problem is like I think the message passing, because you know, especially for like structure modeling, conditioning on a certain structure, we always just be passing the message over like pairs of atoms all the time, and this is like just more difficult.
For example, like a transformer wouldn't make sense here for your problem?
A transformer would make sense. It could work as well, potentially. And these people are trying to do that. Actually, it's called like structure tokenization. I think people, again, moving away from this graph and point clouds is to still use point clouds, but use like kind of transformer.
Yeah.
But I think it's still like, especially on the structure conditioning, like if we operate like in this full box, we're able to capture like all the structure, you know, so like the CNN. So this is a CNN plus transformer architecture, but like the CNN will like capture all the co-correlation between like all the space all together. So this is like the intuition. This is why I would say it's good.
Okay.
And yeah.
Thank you.